So in the last video we met synaptic plasticity and the experimental phenomena of long-term potentiation and long-term depression. So in this video I want to look at the mechanisms of that, which have turned out to be very simple and very elegant. So there are two main questions that arise in consideration of synaptic plasticity. One is, how does the synapse know that it's time to change in strength? So from the point of view of the molecular processes that are sort of sitting there waiting to happen, what is the trigger for them to start making the synapse stronger or weaker? And then the second thing is what actually happens to make that happen. So it turns out that the key to long-term potentiation and long-term depression is a particular kind of neurotransmitter receptor called the NMDA receptor. So there are two broad categories of receptors in excitatory hippocampal synapses. AMPA receptors, which are the normal receptors that respond to glutamate, which is the usual transmitter for fast excitatory transmission. So normally glutamate will bind to an AMPA receptor. The AMPA receptors will open their channels and ions will flow in. But there's another kind of receptor called the NMDA receptor. And this was discovered in the 1980s, and it has some very interesting properties. So if you remember how normal synaptic transmission works, you have the presynaptic terminal and then the postsynaptic dendritic spine, so a little knobble on the dendrite that has the postsynaptic, that has the receptors and all the postsynaptic machinery. So the neurotransmitter is released from the vesicles from the presynaptic side and diffuses across the cleft and binds to the receptors on the postsynaptic side, and then the ions flow in. So NMDA receptors are slightly different because the glutamate binding to the NMDA receptor doesn't immediately cause ions to flow into the cell. And the reason is that the ion channel of the NMDA receptor is blocked by a magnesium ion. So even if glutamate has bound to the receptor and the conformation change has taken place in that protein, ions won't flow. The only way that ions will flow is if the magnesium is pushed out of that channel. And the only way that can happen is if the interior of the cell is depolarized, so it's made more positive or less negative, and that will repel the magnesium ion, and that'll open up the ion channel, and then ions can flow in. And the ions that flow in through the NMDA receptor are the calcium ions. So just to run you through that, two things have to happen for the NMDA receptor to have an effect. The first is that the postsynaptic side has to depolarize and eject the magnesium and unblock the receptor. And then the second is that the glutamate binds to the receptor and opens the ion channel. And then the NMDA receptor channel opens, and it's not sodium ions, but rather calcium ions that flow into the cell through the receptor. And that then initiates some sequence of events that causes the synapse to get stronger. So how do synapses get stronger? So there are several ways, and there was some debate at the beginning about which might be the mechanism of LTP, and I think it's turned out to be true of all of them to a greater or lesser degree. So one possibility is that there could be more postsynaptic receptors. So if you have more receptors, then when neurotransmitter diffuses across the cleft, there are more places for those molecules to bind, and so the sum total of their effects will be bigger. Second possibility is that there may be more presynaptic neurotransmitter. So that's an interesting possibility because, and there is experimental evidence for it that was produced fairly near the start of the LTP investigations, in fact. But there's a kind of conundrum because the conditions for the change to take place are that there's a coincidence between pre and postsynaptic activity. So then the question is, how does the presynaptic side of that synapse know that the postsynaptic thing happened, and therefore there should be more transmitter on the presynaptic side? So there must be some type of retrograde messenger that comes back from the postsynaptic side of the synapse that tells the presynaptic side to make more neurotransmitter. So there's a lot of work trying to identify these retrograde messengers. Third possibility is that there might be a change in the geometric shape of the dendritic spine. So in the picture here you can see the spine has got shorter and fatter. So that means that depolarization in the spine itself will spread more quickly through to the rest of the dendrite and then the rest of the cell. So the same amount of ions flowing in will have a bigger effect on the cell soma. And then finally, synapses themselves may structurally become bigger and more complex. So of all of these, I think the most prevalent mechanism is just simply putting more receptors into the postsynaptic membrane. And we now know that that happens pretty quickly from proteins, receptor proteins that are just floating around in the dendritic spine that are ready to be inserted into the membrane. But also, as we'll see, a slightly more delayed process involves the transcription machinery, so the synthesis of new proteins triggered by genes. So long-term depression, interestingly, also depends on activation of the NMDA receptor. And the only difference is that strong activation causes long-term potentiation. So on the left and the middle here, you can see the release of neurotransmitter that causes long-term potentiation to occur and more receptor molecules to be inserted into that membrane. But on the right, you can see what happens with long-term depression, that a weaker activation still activates NMDA receptors and calcium still flows in, but because it's a small concentration of calcium rather than a large one, the small concentration actually causes receptors to be taken out of the membrane, and so the synapse will get weaker. So a very simple kind of flip-flop type of switch, really. A lot of activation makes it stronger, a little activation makes it weaker. So these processes depend on the synthesis of new proteins for the long-lasting version of them. So as I mentioned, immediately after the LTP has been induced, the receptors are inserted into the membrane, and that process doesn't require new protein synthesis. But that type of long-term potentiation is quite short-lasting because the receptors get taken up again and they disappear. If you repeatedly activate that synapse, then it switches from a short-lasting potentiation, sort of a few hours, let's say, to a very long-lasting form that can last for weeks or sometimes months or even years. We're not sure of the exact possible timescales, but certainly a very long time. So for something to be as long-lasting as that requires activation of genes, because the genes have to be the things that sustain the protein synthesis that keeps us all at the higher level. And so it was shown fairly early on that if you suppress protein synthesis with a chemical called anisomycin, then you'll still get LTP, but it will decay quite rapidly. It'll be only short-lasting. So what do these proteins do? Well, they cause those effects that we saw earlier. So the increase in receptors to be manufactured in order to be inserted into the membrane, the elaboration of the synapse, the change in the shape of the synapse, and so on. So all of these things depend on the formation of new proteins.